Okay, testing, testing. Okay, wait a minute. I check my line. Is it my line okay or no? Wait a minute. Okay. So, okay. So now it's already 8 o'clock. So, okay. Okay, so first of all, I need to say sorry lah because almost 2 weeks, right? I am unable to do the class for Century. The first week, last 2 weeks is because of my uni activities, and then last week is I am sick. I have severe sore throat. I cannot talk at that time. So okay, so hopefully my sounds now just baru sembuh je baru sembuh je so I cannot speak very loud lah for now, so I hope that my sound is clear, right? Okay. Okay, so for today, actually we already finished all chapter 17, right? Inheritance and genetic control, or ectopic, we already finished it. Now we will enter gene technology. So chapter 18 lah, the last two chapter in in Sem 3. So I don't know your school teacher already teach until here already? Some school, maybe your school teacher not yet, your school teacher already finished, whatever, or in progress. Okay. So for today, we will learn about gene technology. So when we talk about gene technology, we will learn about the genetic engineering. So you can see here, it is genetic engineering, not gene engineering. Genetic engineering. Okay, so actually this technology, I think everyone heard about it, right? Even I think in SPM, you are also have heard about it. Right. So what is genetic engineering? So actually, when we call about genetic engineering, actually there are many names lah. Genetic engineering is the former name. Another name you can say that is recombinant DNA technology, recombinant gene technology, gene cloning technology, all these gene cloning technology, recombinant DNA technology, genetic engineering, genetic cloning, all of these are same. Dah banyak nama dia. So genetic engineering when we talk about it is about the technique to modify the genetic material of an organism. So as we guys know, our genetic material in one organism is already fixed. Once it's already fixed for one organism, right? Maksudnya like our genetic material, all of the human, all human must have the same, must almost have the same genetic material. Like chromosome number. Human, all of the human have same chromosome number, right? 46 chromosome only. But maybe computation, some have 45, some have 47. This one, but not. But if humans say that, if you say that human have only 30 chromosome, betul tak? Tak betul. So normally this genetic material, we only modify the genetic material. Normally is modified the DNA lah, the gene sequence. Not modify all the chromosome. We only modify the some of the part of the gene. So why we want to modify it? Because we want to give the desirable trait to certain organism. So for example, some organism, normally this one is very famous technology nowadays, but in Malaysia not very famous yet. But in the Western country, especially in United States, and also United Kingdom, all the Europe country are very famous in this technology lah. So normally this one technology is want to give that certain organism have the desirable trait to organism. For example, like plant. If we for plant, normally if for the crop, jagung, semua makanan-makanan daripada pertanian to in agriculture, all of the plants, normally we need to have the high resistance to pesticide, right? Want to need to have the high resistant to pesticide, insecticide, all the, and also easy to grow. All of that increase the quality, increase all the quality of food, all of these. So this one we will, how to increase it? We can modify the genes of them and then increase it. So normally is give the desirable trait lah. So like contoh, I use human as the example, but for now, as they this technology are not applicable in human yet. Normally is only in animal, plant, and microorganism. But if you want to easy understand, like in human, kalau kami nak menjadi orang yang tinggi atau orang yang kacak, you need to become a handsome boy or beautiful girl. No, you don't need to spend money to do a surgery. You can directly modify our genetic material, modify lah apa gene yang melibatkan awak handsome punya, maksudnya yang ni gene ni menyebabkan awak tidak handsome. So awak modify gene ni, awak akan menjadi orang yang handsome. Macam tu lah. Example dia macam ni lah. Tapi in human is not realistic yet in nowadays. But maybe in future can. But the example is like that lah. So normally the organism that created by using genetic engineering are called genetically modified organism, GMO. So the short form is GMO. So normally if you see the news say GMO, meaning that organism, that plant, that animal, or that microbe already, the gene is already been modified. The gene that they contain is not the original gene already. Dia ada certain gene sudah tukar. Dia ada tukar kepada yang kami nak. Okay, so this is about the history of the genetic engineering. So normally you see here, I just want to mention, normally the genetic engineering is from here. The first GMO produced created through a genetic engineering. GMO tomato. So tomato. If you know that the first pharmaceutical product that produced using the genetic engineering is insulin lah. human insulin. Uh, when you using this technology, we can produce high amount of human insulin compared to the conventional method. In very conventional method, when genetic engineering is not discovered yet, using conventional method, how we get the human insulin? Normally, we get the insulin from the animals one, like cow, like sheep, all that lah. Cow, sheep, elephant, all that animals. But if you use the insulin from that animal, actually they are very risk lah, high risk of contamination or rejection and to the diabetes patient. So when the genetic engineering is discovered and then when it's discovered and then it used to produce the human insulin. So once it using human insulin, it can produce very high amounts, large amount of the human insulin in very short time. So from starting 1982, 1982, the human insulin is not a very expensive pharmaceutical product already lah compared to before 1982. So from starting from this year, normally all the diabetes patient can buy the human insulin in very cheaper price compared to compared to the conventional method, because conventional method you extract insulin for animals, so it's very difficult because you need to isolate the insulin for animal is very difficult, and also there are many healthy issue, healthy problem to the human also, because although it is insulin, but it is come from extracted from different kind of organism, different species, so it is have the health issue lah. So when the human insulin is approved by FDA using GMO. So normally starting from this year, so many scientists starting to discover the genetic engineering technology. So they discover, you see that discover the tomato. Satu tomato ni dia tukar gene dia. Kenapa nak tukar gene dia? Because it need to increase the time of ripening. Normally in traditional punya tomato, ripening time is very low, is very long. You need to tunggu dia sampai tomato dia masak, sangat panjang lah masa dia. So when you use this GMO genetic engineering technique, it can produce the ripening process is very short already. And also increase the quality of the tomato lah. And then you see here normally this one is about and then they apply in many plant lah. So you see here summer squash, soybean, cotton, corn, papaya, potato, canola. So all of these not all of these already the scientists start to modify their gene lah. But for nowadays, not all of the product nowadays is still can be sold in market. Some of them still under the research, but normally in Malaysia, because in Malaysia, due to the ethical issue, so in Malaysia we are not very can see the GMO product in market in Malaysia. Memang tak boleh nampak punya kerana KKM kami, the food scientist, KKM kami tidak akan approve GMO di Malaysia punya. Memang tidak akan approve. Kerana Malaysia ada itu ethical issue lah, banyak lah, either political issue, agama ke semua ni. So Malaysia is not allowed GMO product in market one. They are the act under the Biotechnology Act. Biotechnology Act already mentioned it. So normally GMO product, if you want to sell GMO product in Malaysia, it is very high. You can sell, but the requirement is very very high. Memang sangat tinggi. Until now, until they cannot sell lah. Because GMO product, although it's very good, but also they have some, I say that there are some ethical issue lah. So normally in Malaysia, not everyone can accept it first, and also there are the act government is prohibited this. So so ini Malaysia lah. Saya pun tak boleh cakap apa. So normally this technology you can do in research in Malaysia, but if you want to sell it, for Malaysia now is not allowed lah. Malaysia government is not allowed to do it lah. Okay, normally yeah, and then another one, the most famous one is salmon fish. Kami modify the gene of the salmon, so meaning that this is why we modify, meaning that the salmon can be grow more bigger compared to the conventional salmon. Kalau yang tak ada kalau yang kepada salmon, kalau salmon fish yang tak ada not GMO salmon fish, normally dia kecil je. Tapi kalau kami buat GMO, kami buat GE, genetic engineering, salmon fish dia akan sangat besar. It can grow very in larger size. So until now lah, normally lah. So okay, so this is just the history of the genetic engineering. You can search it if you're interested in it. Sorry. Okay. So this is the very famous, I think it's the latest one lah. Latest technology in United Kingdom. You imagine that when the red tomato combine with the purple snapdragon, apa akan jadi? Ini saya buat research dekat bukan saya buat lah, itu saya ada cari jurnal ni. Red tomato campur dengan purple punya snapdragon, apa akan jadi? You can think about it lah. Okay, so the answer is purple color punya tomato. Tak pernah tempat kan? Purple color punya tomato. Yang ni memang tak akan you will never see it in Malaysia. Memang tak boleh nampak. You can see it only in UK, United States punya supermarket. In United States punya supermarket, mereka ada jual purple color punya tomato. So when they want to create a purple color punya tomato, the research say that purple punya tomato lebih sedap lah. Tak tahu lah, mereka cakap punya lah, saya pun tak makan. Mereka cakap purple punya tomato lebih sedap compared to yang ni, compared to yang black tomato. Mereka cakap macam tu lah. But in Malaysia memang tak akan jual ni. If you want to try it, please buy your ticket, go to United States and then buy it lah. In United States there are many product already is GMO lah. It's very common nowadays in United States, but in Malaysia, sorry, not yet. Malaysia not yet because there are many issue in GMO nowadays. Okay, so ini cerita in fact. So now we go to the main point, that is the main stage in genetic engineering. So what are the genetic engineering technique? Macam mana kami nak buat macam ni? Macam mana kami nak using tomato dan juga ni campur jadi tomato purple ni? Macam mana? Ini way dia, inilah 5 main stage this. So there are 5 main stage. So these are the steps, that we need to use to create a recombinant, create the GMO product. So ini boleh baca. Okay, we will go to one by one. Okay, so this is the overall process. So in general, normally in this genetic engineering, we will use certain things. The first thing is host cells. So the host cells, the most common host cell we will use is E. coli, bacteria. Bacteria cells, E. coli lah. E. coli akan digunakan sebagai host cell. And then because why we use E. coli? Because with the E. coli we have plasmid. Plasmid, it will become act as the vector. Ini akan menjadi vector. So actually plasmid in bacteria, it is the DNA molecule. It is a DNA molecule, but within the bacteria, it is not play the role in it does not play the role in the in the play the role like the genetic, like the chromosome one lah, like the play the characteristic, like play the characteristic of the bacteria. But normally plasmid is play the other role in bacteria. But anyway, the plasmid is the main vector that we use. Okay, and then after that we will have the gene of interest. Kami kena ambil gene yang kami nak. Because I already say genetic engineering is modify our gene. So memanglah kami kena nak ambil gene yang kami nak. Apa gene yang kami nak? So for example, kalau awak nak gene yang handsome, jadi handsome punya, awak ambil lah. Gene yang boleh menyebabkan awak handsome, awak ambil itu gene. Lepas tu awak ambil gene ni, lepas tu insert the gene into this plasmid. Okay, so this plasmid now it already contain gene yang menyebabkan awak handsome. Jadi awak menyebabkan awak handsome. So when this plasmid is already contain the gene of interest, it will into the host cells again, and then the host cells will be undergo cloning. It will be clone, and then produce more and more of the host cells. So this host cell then it will express the protein. So normally this gene, as we know that gene expression lah, you already learn gene expression. So maksudnya dia akan hasilkan protein yang kami nak lah. So example is insulin lah. So it will express more high amount of insulin at here, and then this insulin will be used in many in the pharmaceutical field lah. So it will be helpful to the diabetes patients. Not only that, it also can be used to transfer this gene into the plant, like this tomato. When you already create the recombinant punya plasmid, you can transfer the gene, this plasmid, into the tomato, into this tomato. Transfer to this tomato. After that, it will produce it. After that, cucuk dia akan menjadi tomato yang purple color macam ni lah. So this snapdragon is the gene of interest. Kami akan ambil gene daripada snapdragon ni, lepas tu masukkan gene ni kepada tomato ni. Ambil gene ni, ambil gene snapdragon, lepas tu masukkan gene kepada tomato. Lepas tu anak-anak tomato ni akan hasilkan tomato bukan yang merah punya lah, akan hasilkan yang warna ungu, warna purple lah, akan hasilkan warna purple punya tomato. So this is just the overall process. We will see one by one of this process. Semua proses we will see one by one. Kami awak kena refer one by one. Okay, so stage 1 is restriction enzyme. Kami ada restriction enzyme, meaning we will see here obtain copy of required gene from DNA donor cell. Awak kena obtain required gene, meaning gene of interest lah. Mana awak ambil gene of interest? Mana awak ambil? From donor cell lah. Cell yang kami nak. So kami nak DNA ni. Okay. But DNA, we not use all DNA because we know that DNA is very long. We only use certain DNA. Certain part of the gene only. So, macam mana kami nak tentukan certain part gene tu? Macam mana? So, the main role is using restriction enzyme. Because this enzyme is used to cut DNA molecule at the specific restriction site. Dia akan potong DNA molecule tu. So, dia akan potong dua benda. If you see here, you see here, dia akan potong plasmid punya gene dan juga potong DNA punya donor cell punya DNA. Akan potong dua tempat. Satu tempat plasmid. Because it want to insert the gene, right? The gene of the donor cell dan insert dekat sini. So maksudnya satu itu restriction enzyme akan potong dekat sini, dekat plasmid, dan juga dia akan potong juga dekat DNA punya donor cell. Okay, so the first step actually, the first step you need to isolate the plasmid and also the DNA first. This is the first step. You need to isolate plasmid and DNA first. Plasmid of the E. coli and also the DNA from donor cell. After that, you will use the restriction enzyme to cut the DNA molecule. Okay, macam mana dia nak cut? Bukan macam mana. Where he cut? Where this restriction enzyme cut? Because dia bukan potong sesuka hati punya. Dia bukan potong dah potong sini, potong sana punya. Bukan. Because we know that enzyme is highly specific. Excuse. It is highly specific. So, maksudnya dia ada tempat yang spesifik untuk dia potong. Hanya boleh potong-potong dekat tempat tertentu saja. So, you see here, so normally there are many types of restriction enzyme. So, normally in our syllabus, we only learn 2 enzyme, which is echo R1 and also small one. Okay, so echo R1 and also small one. So if you see that these 2 enzymes, you need to remember it, and also you need to remember the vagonization site, restriction site. Meaning echo R1 will only cut GAATTC. And small one only cut CCC, GGG. Awak memang kena memorize punya. Tak ada pilihan lain. Ini 2 je. Sepatutnya ada banyak. In university, awak kena memorize semua ni tau. University lah. University memang kena awak memorize semua ni. But not only dia banyak lagi actually nak memorize all. But in STPM, MPN sangat baik hati. Awak hanya belajar 2 saja. Echo R1 and also small one. Yang lain, awak boleh tengok, tak perlu hafal. But echo R1 and small one, you must memorize it. Echo R1 is GAATTC. Small one is CCC, GGG. Small one is more easy, CCC, GGG. Echo R1 macam complicated lah. GAATTC. Memang awak kena memorize. Kalau ayah saya sebut echo R1, memang GAATTC. Small one, CCC, GGG. Semua ni lah. So, these 2 enzymes, so echo R1 only will cut the site. When it vagonize, dia akan vagonize. Akan pergi cari. Oh, dia cari GAATTC. Okay, dia ada potong terus. Okay, small one, dia akan cari DNA. Oh, ada CCC, GGG, akan potong terus. Dia potong punya. Dia akan potong kalau dia ada nampak sequence ni. Kalau dia lain, selain 2 ni, except these 2 enzyme will not take action at all. Dia tak akan buat any action. So, this is the restriction enzyme punya function lah. So, macam mana dia potong? Dia bukan potong sesuka hati. Dia ada potong. If you see here, meaning that GAATTC dia ada slash here, right? So, meaning that dia potong dekat G and A punya part. G and A punya position. Dia potong dekat sini. And for you see here small one, dia potong dekat sini kan. Tengah-tengah CCC, GGG, tengah-tengah. Dia akan potong tengah-tengah punya. So, macam mana dia potong? Okay, so now, if you see here, awak boleh nampak yang paling penting, this one is echo R1 punya mechanism. Ini adalah small one. So, if you see here, echo R1 punya, selepas awak tengok, ini plasmid, right? This is the plasmid, and then this is the restriction site. Excuse. Restriction site. Okay, you see that GAATTC, and then you see that GAATTC. Okay, so you see there, totally same. Now restriction enzyme dia akan recognize. Okay, dia dah carilah. Inilah tempat yang saya akan potong. So dia akan potong dekat mana? Dia akan potong dekat G dan A punya. Dia akan potong dekat sini. Akan potong dekat sini. Lepas tu dia akan pecah lah. Akan pecah. So, you see here, dia akan pecah macam ni. Tengok. So, dia akan produce the end we call as sticky end. Dia akan potong macam ni punya. You see here. This end we call as sticky end. So, echo R1 will produce sticky end. Dia potong. Lepas tu dia akan jadi end yang macam ni. But for small one, you see here, Lepas dia potong dekat tengah-tengah ni, dia akan produce end macam ni. Dia akan keluar satu punya. You tengok ni, dia akan teguk keluar punya kan. Dia akan keluar dekat sini. Ada keluar TTAA dia macam terkeluar kan. But for ni, dia tak keluar punya. Memang dia akan sangat shining punya. Shining punya. Macam yang ni tak shining kan. Yang ni memang shining punya. Dia potong terus. So yang ni we call as blunt end. BLUNT. Blunt end. So this is the two end that produce from these two different enzymes. Echo R1 will produce sticky end, small one will produce blunt end. So if you see here, this is more clear lah. Sticky end, awak tengok memang dia ini DNA sequence right? DNA sequence dia memang macam ni. So if you see here, when it recognize the GAA-TTC, the echo R1 recognize the GAA-TTC, dia akan potong dekat sini. GA dan juga GA dekat sini. Anti-parallel lah. Potong sini, dekat potong sini. And then it will produce the end like this. Memang kami panggil ni call as sticky end. Yang ni end, we call as sticky end. So normally, and then echo R1 also can mix and anneal this anneal mix and anneal this product lah. Ini kami later tengok. But the most important is I want to show you is the end that produced by the echo R1. So please memorize it. Echo R1 is GAA-TTC. Dia potong dekat between G and A punya. Lepas tu it will produce sticky end. And then this one is blunt end, small one. So small one is CCC-GGG. So dia akan potong dekat tengah-tengah punya between C and G. So dia akan potong tengah-tengah. Dia macam potong kek lah. Potong directly potong je. Lepas tu dia akan produce the blunt end. You will produce the blunt end like this. Already two separated lah. Separated strand already. So this is the restriction enzyme that we use. So echo R1 is GAA-TTC. So small one is CCC-GGG. So memorize it. Awak memang kena memorize punya ini tak ada pilihan. Okay. Yang ni pun sama lah. Yang ni pun sama lah. Masa-masa lah. So normally the first step is to digest with restriction enzyme. So normally dia akan pergi to recognize it, the restriction enzyme. Echo R1 akan pergi recognize the site, and then dia akan cut the DNA, and then after that it will produce the sticky end, and then it will mix together. So this mixing, if you see here, this mixing is mixing between the gene of interest and the plasmid. So maksudnya macam ni. Yang ni. Dia akan producekan gene of interest yang purple color punya. Purple color punya dia sudah potong gunakan restriction enzyme. Dia sudah potong. Lepas tu plasmid pun akan potong. So the cut region of the plasmid will be inserted by this gene of interest. So dia macam ni lah. So you assume that this is the plasmid, this is the plasmid, and then this is the gene of interest lah. And then after that, the gene of interest akan pergi ke sini untuk mix dia to form the recombinant plasmid. So like that lah. And then after that we will use another enzyme called DNA ligase. So this DNA ligase is to join the DNA strand. So if you know that because already potong, so normally there are no bond at here. So want to join the nucleotide between here, you will need to use DNA ligase, same as the mechanism in DNA replication lah. So the mechanism is same in the DNA replication. You can refer it about the mechanism in DNA ligase. So that's all for the first step. So pertama kali kami isolate plasmid, isolate DNA, lepas tu potong potong the DNA, cut the DNA using the retrievation enzyme. So there are two retrievation enzymes you need to understand, echo R1 and also small one. So echo R1 is the restriction site is GAA-TTC, and then small one is CCC-GGG. So after you cut the DNA, cut the DNA in the gene of interest and also the plasmid. So this gene of interest will be inserted into the plasmid. This gut plasmid. So it will insert this anti region of the plasmid lah. So it will form the recombinant plasmid. This one we now call as recombinant plasmid. Okay, this recombinant plasmid later what will we do is it will be transformed to form the recombinant bacteria, recombinant E. coli. So this is the next step lah. Okay, so this one is the terms that you need to know it. This term is called pendidomic sequence. So what is pendidomic sequence? Normally it is the sequence varies from 5 pi to 3 pi and 3 pi to 5 pi direction, or two anti-parallel strands show sequence similarity. So if you see here, awak baca dari 5 pi to 3 pi or 3 pi to 5 pi, ini direction awak boleh nampak dia memang sama je. DAA-TTC, GAA-TTC. Memang sequence dia sama. So if you see here pun sama. This is the two complementary strand lah. In the single strand also same, you see that TTACG. So at the another end is TTACG. So memang dia bacaan nucleotide dia memang sama. So this one we call as pendidomic sequence. This sequence is very important in genetic engineering, especially we need to recognize the retrievation site of the enzyme. So all of the retrievation enzyme, retrievation site of the DNA of the DNA strain totally are pendidomic sequence. You cannot say the retrievation site is not a pendidomic sequence. If not pendidomic sequence, memang tak akan the retrievation enzyme will not cut it lah. So it must be pendidomic sequence. So this is the term you need to know it. Okay, now we go to vector. Okay, so kami guna vector kan? Kami dah mention we use vector. Okay, now we have a question. Why we want to use plasmid as a vector? Kenapa kami guna plasmid ah? Kenapa kami tak directly guna chromosome ah? Why? Or why we cannot use human punya DNA? Why we need to use plasmid? And also another one is bacteriophage. Why you use only these two? Why we cannot use yang other like chromosome? Kenapa tak boleh guna chromosome ah? Kenapa tak boleh guna DNA ah? Kenapa tak boleh? Why we want to use this? So there are the reason. So why we need to use the plasmid? So these are the reason. There are 5 reasons. First reason is small. Normally the plasmid, the sequence size is very small. Is very small, and also the size is very small. So meaning that it is very easy to purify and also insert into bacteria cell. Compared to you directly affect the bacteria, the DNA of the bacteria cell is more difficult, because we know that the DNA in bacteria cell, although it is narrow, but it also very long like the human one. But compared to human, it's more shorter lah. But in bacteria, even the bacteria alone, it also have the very long DNA. So very difficult. You want to purify it is very difficult, and also you need to insert again into the bacteria cell, also very difficult. So this one, so why we cannot use chromosome is this reason, because chromosome is very large and complex and not easily to purify and insert. Okay, this first reason. Another reason is DNA sequence, because plasmid we know that it is very small, so meaning that it very easy to know the sequence of all plasmid. So for 90 cell they already know the total sequence, all these sequence apa itu DNA sequence semua ni scientist sudah tahu. Memang sudah tahu. So meaning is very easy to manipulate. Compared to when you isolate bacteria cell, isolate bacteria cell, you know that the DNA is very long. Wah, apa nak baca DNA sequence tu nak take time take very long time. And compared to the compared to plasmid is very small. So you need to make a research to know, understand the DNA sequence is more easy compared to when you use the chromosome. Another one is number three. This one better we will use. It has the selectable gene marker. So this one is about the checking process that we will learn later. So normally this one is an antibiotic resistant gene and allow bacterial cell contain plasmid to be isolated. So this one I will explain later. Another one, many unique restriction site. So plasmid, they found that within the plasmid, there are many unique restriction site. So you see here, awak tengok ni. So you see here. So for this, actually there are many restriction site at here. Yang ni. Yang purple color punya is the restriction site. So actually there are many restriction site at here. And also each site is very unique. Meaning that you can use different type of restriction enzyme to cut it. And only one that enzyme can cut that part only. Like if you use that echo R1 hanya boleh cut dekat sequence apa? CAATTC right? GAATTC right? GAATTC. Dia akan boleh cut GAATTC right? Small one is SSCCC GGG right? So there are many restriction sites. So it is very flexible and easy to encoding. So more flexible. You don't need to sakit kepala. Oh, kalau plasmid ni tak ada. Sorry. If this plasmid does not contain any restriction site, this plasmid cannot use. No. Actually, there are many unique restriction site. And when you use that restriction enzyme, you don't need to sakit kepala. Dia akan cut dekat tempat lain. Maybe dia akan cut dekat tempat lain. Tak akan dia. Dia akan cut. It only cut in that part only. Like it will cut like echo R1, it will only cut at here only. Memang dia akan potong dekat sini punya. Yang lain memang dia tak akan potong punya. Tak akan potong. Because only this part, it contain the restriction site of echo R1. So it will only cut at here. Yang lain the part, tak ada. So dia tak akan cut. So echo R1 will cut at here only. So this is what we call as very unique restriction site. And also rapid replication. You can, the plasmid can replicate rapidly in very short time. So compared to you use the chromosome all the very complex one, very large DNA punya, not very easily to replicate lah. Because it's very long, so it take time. So but the plasmid is short and also small, so it very easily to replicate. So this is why we want to use the bacterial plasmid and the characteristic of the plasmid that have lah. So this is one of the example that we use PUC19. So there are many one of the example PUC19. Okay, another type of vector that we use is bacteriophage. So this bacteriophage normally we use when we want to clone the genetic sequence is very long compared to plasmid. Because plasmid normally it only can clone the gene sequence normally is about 5,000 to 5,000 base pair only. 5,000 base pair only. But for this bacteriophage normally can up to 10,000 base pair. Dia boleh up to 10,000 base pair. So for this one is normally when you want to clone your gene of interest is very long very long. So if very long you need to use bacteriophage. But the most common one is we use bacterial plasmid. But bacteriophage is where we want to clone very long, more complicated of DNA interest. So bacteriophage also we can use it. So why we need to use it? So normally there are also same reason as the bacterial plasmid, but just that also have the same reason as the plasmid that we use. It has a known DNA sequence, replicate independently accept last piece of foreign DNA efficiency in DNA transformation. So the reason you cannot say that bacteriophage is more small lah, because bacteriophage is one type of virus. Bacteriophage is one type of virus, so you cannot say that bacteriophage is small lah. So it has a no DNA sequence. You already know the sequence of DNA for the bacteriophage, and the unique is it can replicate independently compared to the plasmid. Compared to this plasmid, normally plasmid it replicate based on the host cells. Maksudnya satu host cell dia hanya ada satu plasmid je. Satu plasmid je. But in this one, it replicate independently, meaning that in one host cell, not only or not necessarily only contain one bacteriophage, it can produce many bacteriophage in host cells, like E. coli. Normally in one E. coli, it can produce 100 bacteriophage, maybe 100 bacteriophage. So normally bacteriophage is more efficient lah, compared to plasmid lah, because we are compared to bacteria lah, compared to plasmid. Why? Because one bacteria only contain one plasmid, and then this one bacteria maybe can contain 100 bacteriophage already. So yeah. So that is why that we say as it can replicate independently, and then it can more efficient. So it can produce more product compared to the plasmid. And also accept last piece of foreign DNA. This one I just mentioned lah. And also it is efficient in DNA transformation. So it is easy inject of their chromosome into the host cell. So this is the reason that we need to use in the bacteriophage. Okay. Okay, so this is the characteristic of the vector we use. So any question? Okay, no question. Okay. Now we go to the injection process. So after we already cut so you see this diagram, we already say that we already cut the plasmid, right? We already cut the plasmid and also the DNA interest. So this one this one is the isolation. So you already isolate the plasmid from the E. coli, and also isolate the of the DNA, isolate DNA from the gene of interest. I isolate the gene of interest from the donor cell. So okay, kalau dah dua ni, we already mentioned we will use the restriction enzyme. So for here we use echo R1, and then we use echo R1, kami potong dekat sini, potong ni dua tempat ni, and also for DNA kami potong. So it will produce the sticky end. So you will see here clearly complementary sticky end. So after that, this gene of this DNA, it will inserted back to the plasmid. So how about this plasmid? The original plasmid and also original gene. So yang ni kami tak mau lah. Kami tak mau. Kami hanya mau yang ni je. Ini gene of ini DNA, the cut DNA only, and also the cut plasmid. After that, we join it together. We insert this DNA, this cut DNA into this cut plasmid. We will form the recombinant plasmid. After that, we will use the DNA ligase to join the bond, to join the DNA nucleotide between them lah. So this is all the explanation you see here. Then the restriction site within plasmid is cut with the restriction enzyme. So then ini paling penting lah. The plasmid will join with the donor DNA restriction by DNA ligase through the joining of focal receptor to form the recombinant DNA. So this one will form the recombinant plasmid lah, or we call as recombinant DNA lah. Okay, so ini injection process lah. So hopefully you guys understand. So now we already formed the recombinant plasmid already. We already modified the gene sequence of the plasmid. Plasmid ni kami dah modify lah. Yang originalnya sepatutnya ni, tapi kami sudah insert the gene of the interest yang kami nak. Maksudnya macam ni lah. For this, ini yang boleh menyebabkan handsome. Ni gene boleh menyebabkan handsome. So, yang ni sudah ada trait yang boleh menyebabkan awak handsome. So, if you got this gene, awak akan menjadi handsome. Like this lah. Meaning dia lah. Okay. So after that, we will go to the transformation. So we know that after the process recombinant plasmid is formed, we will insert this plasmid back into the host cells. So the host cell is E. coli lah. Okay, so the process we call as transformation. Okay, so normally the host cells will have this general characteristic. So hostile, not necessarily is using E. coli, but the most one is using E. coli lah, bacterial cell. So normally the characteristic of the host cell it can be able to receive recombinant DNA, maintain the structure of recombinant DNA from one generation to other, meaning that these host cells, after it produce more generation, the recombinant plasmid also is very stable. Not say that after it produce the next generation punya hot cell akan produce anak daughter cells, meaning that the recombinant plasmid will be affected. Tak boleh. Kalau macam tu, yang tu that cell cannot be as the hot cell. So the hot cell must not maintain the stability of the recombinant plasmid. And then of course, it be able to amplify the gene product from recombinant DNA lah. And then the most common host cell is bacteria. Why we use bacteria? Normally, it's easy to be isolated, and also the bacterial cell cause repeatedly replicating. So bacterial cell can be replicate rapidly in any foreign cell. Dia akan ada effect punya. Dia akan ada ini plasmid ni dia akan ada apa-apa effect kepada bacterial cell. Kerana kami tak sentuh the genetic, net DNA of the bacteria. Semua metabolism process, all the metabolism process are still can be maintained. Just that we only modify the gene of the plasmid only. But the plasmid actually does not affect does not play the role in metabolism process. So bacteria cell akan survive macam normal je. Just that it will produce the desired product that we want lah. So kami macam ni, in normal bacteria cell dah tak akan produce the trait that can make us handsome. So from now, it already can produce the trait that can make us handsome already. Yang maksudnya lah. So maksudnya health cell ni it can be replicate rapidly. And also the process of transform recombinant plasmid into the host cell is called transformation. Okay, so in general, there are two types of transformation process. How we transform the recombinant plasmid into the host cell. First one is using chemical transformation. We using the chemical treatment. Normally is sodium chloride, and also you using the electroporation. Using electric shock. Electrical punya method. So okay, so normally this one is when you use the chemical. So normally the chemical that we use, there are many chemical lah, but the most chemical we use is calcium chloride. You also can use magnesium chloride. Other one magnesium chloride, and also you can use the RBCl. So normally these are the mechanisms. So how it affect the host cell. So normally this one is known as the competent cell. So competent cell is meaning that cell does not have any plasmid, because we already isolate plasmid, right? So this cell, the host cell, we now know as the competent cell. So this competent cell does not contain any plasmid one. It will stand by to receive the recombinant plasmid. Okay, so as we know that bacterial cell contains cell wall made by the peptidoglycan that you already learned in semester 1 punya structure, prokaryotic structure, we know that it contains the cell wall and also capsule. So it is not easily, but not very easy, for N material, you want to insert the for N material kerana dia ada bacterial cell wall halang dia. So you need to need to do something to instable the cell wall of the bacterial cell. Awak kena buat sesuatu benda to make sure cell wall dinding dia macam tak stable, so that the recombinant plasmid can be transformed into it. So there are two method lah, chemical and electro. So ini chemical lah. So normally chemical is that when there are the chemical at here, so normally the calcium chloride, it will neutralize the negative charge of the phospholipid bilayer and DNA. So we know that the DNA carry negatively charged, and also phospholipid also carry negatively charged. So that this calcium chloride, once it enter at this region, the calcium chloride will be interact. The calcium ion, calcium ion will interact with the negatively charged, cause the plasma membrane and the DNA is neutral environment. Kerana kami pun tahu when it carry the negatively charged, meaning that the DNA recombinant plasmid memang tak boleh masuk because it will repel. Right, it will repel to each other. Negative same charge will repel each other. Memang tak boleh masuk. So when the calcium ion is bind to the phospholipid bilayer ni, and also bind to the the DNA, so it will become the neutral. So it already become unstable. Now the recombinant plasmid can able to enter the bacterial cell wall and then go to the inside lah. So the recombinant plasmid able to transform into the bacterial cell. Okay, so ini principle dia. Okay, and then this is the experiment process. Normally yang paling penting here, allow the cell begin to grow and ensure that the antibiotic resistant. So normally this is the process. Yang paling penting, you need to have the heat shock. Not only using the calcium chloride, not only pour the calcium chloride and then that's all. You also need to heat shock because you need to permeabilize the membrane cell. Also need to make the membrane cell is more permeable lah compared to yang ni. Even though if it is neutral, but you know that phospholipid bilayer cannot, the original punya phospholipid bilayer memang tak boleh bagi ni masuk punya kerana terlalu besar. So kami kena buat heat shock make the plasma membrane is not very tightly to each other, memang loosely to each other, and then it more permeabilize so that the recombinant plasmid can enter it lah. The heat shock is very important. Okay, yang ni is using the electrochemical punya. So normally this one is more easy compared to chemical because you don't need to use chemical, you just directly using the electrical shock. Directly using the electrical, using the laser ke apa high electricity in very short second. After that, when the then it will cause the plasma membrane, the cell wall is permeabilized. After that, it will directly, the recombinant plasma directly will be transformed into the host cell, E. coli. So electrical shock is more easy compared to chemical, but electrical shock is more expensive compared to using the chemical way. Okay, so this is the transform. Okay, after the transform process, actually the genetic engineering process is already finished. Maksudnya you already finished already. Actually it's already finished. Now after that, after genetic engineering, after when it transform the bacterial host cell already contain the recombinant plasmid, then it will directly clone it to produce large amount of bacterial cell that contain this recombinant plasmid, and then it can be expressed to produce the desired trait. Actually like this only. But as a professional scientist, you cannot end like this, because scientists actually a very not a smart scientist one. Scientist is memang akan mengalami kekalahan punya. Memang awak akan mengalami kekalahan punya. So, we cannot guarantee that once we do all this process, that the culture solution, the solution that we get totally contain recombinant plasmid. Awak can you say that totally inside your culture solution contain recombinant plasmid? Bolehkah? Siapa boleh confirm? Kerana awak tak boleh nampak pun. You cannot see by your naked eyes. You only can see cannot see in your naked eyes. Even in microscope pun awak boleh tak nampak. Because you don't know that ini betul tak recombinant plasmid. Maybe it is the normal plasmid, or even though it does not contain plasmid. So actually inside the culture solution, after you do all these steps, inside the culture solution, not all the cells, not all the bacterial cell contain the recombinant plasmid. So what we do is we need to do screening. Memang kena screen punya. Do a final checking. Like you do examination, right? Examination, right? After you finish all the question, you need to do what? You need to check, right? Check your answer, correct or not, correct or not. Like number one, ABC. Number one, I think it's correct. Or normally, I think it's here. I say question, I tengok macam pelik je, saya tukar-tukar. So you need to do a screening, same as the genetic engineering. You need to do a screening because you cannot ensure that all of the cell in the culture contain the recombinant plasmid, or even the cell does not contain any plasmid. So we don't know that. So we need to do that screening. So how we do that? Normally we will use the most famous one is blue white screening. Okay, so normally what is the blue white screening? So this is the result yang kami dapat. Ini kami dapat punya. Apa itu? Normally when you after when you you will see here, normally you will see here, when you do the recombinant plasmid, when you already enter transform into the host cell, so this culture solution will pour into the alga medium. This alga medium will contain two substance. First substance is ampicillin, one type of antibiotic. Another substance is called agar. Agar and ampicillin. Okay, what are these two functions? So these two substance is very, very important in screening process. Okay, agar, so I go to the ampicillin first. So normally ampicillin is based on the resistant antibiotic resistant gene that found in the plasmid. So this is what we say that one of the characteristic here. Selectable gene marker. We need to select the plasmid. So in the vector, the plasmid, normally the plasmid will contain the ampicillin resistant gene. So not necessarily will use ampicillin. Actually, you can use other type of antibiotic, but normally in your syllabus, you just want to know it will use ampicillin only. Ampicillin. So normally, if you see this plasmid normally almost all the plasmid will contain the ampicillin resistant gene. Meaning that when the bacterial cell contain the plasmid, even though you give the ampicillin toward this bacterial cell, that bacterial cell will not die. As we know that bacterial cell will die, antibiotic can kill the bacterial cell, right? But if the bacterial cell contain plasmid, and also this plasmid contain the ampicillin resistant gene, meaning that ampicillin, although it act as an antibiotic, but it cannot kill that bacterial cell. Dia tak akan bunuh bacterial cell punya. So when using the ampicillin, so when there are the colony occur, so you see here, kalau ada colony occur, tak kira dia blue or white, ada colony occur, meaning that the cells contain the plasmid. Cell ni ada plasmid. So meaning that these cells either it is already successful be transformed or this cell is already original contain the plasmid. Original punya plasmid. So yang ampicillin ni, you just want to make sure that transformation is occur. Yang kepada host cell yang tak ada transformation punya, macam ni competent cell, if after transformation still competent cell is exist, so normally it will die lah. Tuang ampicillin je, bacterial cell ni akan die lah, kerana dia tak ada plasmid ni, dia tak ada plasmid. So tak ada plasmid, so dia akan die lah. Ampicillin akan bunuh bacteria tu. So normally it will only give two results, either it had colony or no colony at all. So if no colony at all, so yang ni saya rasa saya tulis salah. So normally if you want to have the desired product, so it must have the colony. So the colony. Undesired is no colony. Okay. So when you have the desired product, when you have the colony, meaning that the plasmid is present. And then when there is no colony at all, tak ada colony langsung, maksudnya plasmid tak ada, memang dia kerana dia dibunuh oleh ampicillin. Ini just the first step, ampicillin je. But we still don't know is it this cell contain recombinant plasmid or not. Because it may be that the plasmid is not recombinant, it's the original plasmid. Kami tak tahu. So now we go to what you would call as Bouvard screening. That the X-gal play the role lah. So what is X-gal? So normally X-gal is one type of carbon, is one type of substrate. So when you go to the structure of plasmid, you can see that there are the LATZ gene at here. LATZ gene. LATZ gene. Macam masa-masa je, macam ada pernah dengar kan, you eat before kan, mana heard it, think about it, so you heard it before it, LATZ gene. So we know that LATZ gene will express beta galactosidase, one type of enzyme. So this X-gal when it contain the beta galactosidase, it will hydrolyze the X-gal, and then it will produce the blue dot colony. Okay, this is first situation. Ada dua situation. First situation is when there are the LATZ gene occur in the plasmid, and the plasmid, it will express the enzyme called beta galactosidase. After that, it will hydrolyze the X-gal to produce the blue colony. Because it will produce the one type of product, and then this product will produce the blue colony in the medium, agar medium. The first situation, if the plasmid does not contain any LATZ gene, so it will not hydrolyze the beta galactosidase. So the X-gal will not be hydrolyzed, so it will produce the white colony, because X-gal is not hydrolyzed, so no product form, no the blue product form, so only white colony form. Okay, so my question is, what are the desired colony that we want, blue or white? So you see here, we say that white is desired colony, meaning that white is the colony that contain recombinant plasmid, meaning that white color is the cells, E. coli cell yang kami nak. Blue kami tak mau. Kenapa? Apa LATZ gene ni play what role? Apa peranan dia? So normally LATZ gene play the role in the retrievation site. So normally in let Z gene, it will contain the restriction site will be exist in the let Z gene. Meaning that the restriction site let Z gene here apply. Ini let Z gene right. Meaning that dekat let Z gene dia ada restriction site ni. So if you successfully cut the let Z gene, maksudnya let Z gene ni dia tak apa, dia dah jadi bukan let Z gene lah. Dia dah sudah potong, the genetic sequence is already altered, so now the recombinant plasmid will not contain the let Z gene one. Dia tak akan ada let Z gene punya. So that's why that white colony is the desired colony that we want. Because it already does not contain any let Z gene, meaning it only contain it is already exists recombinant plasmid. So this product, this cells, white colony is the colony that already contain recombinant plasmid punya cell. Yang blue punya, all this blue, it contain plasmid, but that plasmid does not contain, it does not recombinant plasmid. It is the original plasmid or plasmid yang datang dari mana-mana lah. Saya pun tak tahu datang dari mana. But blue one is not, kami tak mau. Kami nak white. Okay. So this is the screening process. Ini screening dia lah. So, macam mana screening? First step is using ampicillin. After that, you using the X-gal, and then it will produce the white colony. White colony and blue colony. White colony, kami nak. Blue colony, kami tak mau. Kenapa? Because the original plasmid, the restriction site is within the let Z gene. And then if let Z gene is present, it will express beta-galactosidase. Beta-galactosidase will hydrolyze X-gal to produce blue colony yang kami tak mau, because when it produce blue colony, meaning let Z gene is present. So let Z gene present meaning dia plasmid ni adalah original punya. Tak ada, memang tak ada recombinant. So kami tak mau, kami yang nak putih punya. So putih is a gene, is the cells that already contain recombinant plasmid. So we know, so from here we know that kami nak putih punya. Yang biru punya kami tak ambil. Kami hanya ambil yang white colony one. So after we know that, next we can directly go to cloning process. Okay, so this is blue white screening is one type of screening. Another screening is this NA probe. Nuclear nitrocellular filter probe. So this one is more complicated, complicated to understand compared to this, but never mind. I don't know you guys tak dengar tak? Because I think mostly your school teacher will not teach this, but they just put in the slide. Yang ni, this is the university level one lah. I don't know. Actually, I don't know STPM ada belajar tak. Even, my STPM is not belajar. Saya tak pernah belajar ni. Because school teacher not teach. Tapi in MPN syllabus dia tak mention. So, saya tak tahu ada tak. So, normally, yang ni, normally I just briefly explain lah. Normally, this one use the DNA probe. Kami buat itu perbandingan punya. So normally awak akan guna ini nitrocellular filter. Awak filterkan, awak akan macam photostat lah. Photostat the DNA punya structure dekat ini filter. So maksudnya filter ni dia akan ada DNA sequence dekat sini lah. Dan macam buat awak dekat koloni dekat dia lah. Lepas tu awak guna ini DNA probe. So normally this DNA probe is the label probe. It already contain label probe. So ini label probe is the probe that we need to plot probe that we already know that what sequence we want. After that, it will directly hybridize with any complementary DNA. So it will hybridize with hybridize with the this biblical filter, ini yang ni nitrocellular filter. So after that you go to the X-ray film. So when you pass to the X-ray, so the DNA that bind with this DNA probe, dia akan keluar itu white dot lah. Dia akan awak under the X-ray film, X-ray film awak akan nampak itu white dot punya. So yang ada white dot punya maksudnya adalah koloni yang kami nak. And then this, because this is the filter paper, ini filter paper. So you can make a comparison with the bacteria colony agar black. So maksudnya filter paper ni dia macam copy lah, dia macam awak copy punya DNA information to this this filter. After that dia akan hybridize with this DNA probe. So kepada koloni DNA yang ada recombinant plasmid, dia akan ada certain DNA yang kami nak. So this DNA akan bind dekat DNA probe ni. So after that dia akan run dekat X-ray. So dia akan form the dot lah. So yang form the dot ni adalah dot yang kami nak. Okay, and then awak boleh bandingkan dengan bacteria colony plate ni. So awak beza lah. Beza awak boleh macam awak tengok, kerana ini transparent punya, awak boleh tengok directly. So awak dari phone ni awak boleh tengok, boleh nampak koloni ni 1, 2 and 3. These 3 colony are the desired colony we need. Yang others is not a desired colony. So this another screening process lah by NA probe. So ini overall dia. For if you are interesting, you can search it. Okay, now we go to the cloning process. So cloning process, actually in our syllabus there are 2 cloning process that you will learn. Either you using the culture medium. Either you using the culture medium, or either you using the PCR reaction, polymerase chain reaction. So culture media is the conventional method. So after you know that the desired colony, this white colony, you know that you want to clone this. So you take out this colony, go to another culture medium, either enriched neutral medium, and then you incubate it lah. Awak incubate dia, make the DNA more replicate. The DNA will be replicated more and more high amount of number, so that the media after the several days, that media only contain the E. coli that contain the recombinant plasmid that we want. So like the tadi punya example lah. So after you clone that, you go to that, the culture medium only contain the gene yang boleh menyebabkan kami handsome. So awak maksudnya memang macam itu medicine right. Awak minum saja awak jadi handsome, macam itulah. So this is the culture medium lah. So this is the conventional method and also the easiest method lah. Another method is modern method, either you using the polymerase chain reaction. So the polymerase chain reaction normally is use the amplification method. So normally if you know that you already have the gene of interest, so normally you can isolate the DNA and then replicate more and more like this. Like this. So ini salah satu method dia. So normally DNA polymerase chain reaction is more complicated. So normally we see here, DNA replication contains 3 main steps. The first step is we need to denature the double-stranded DNA into single-stranded DNA, and then we will have the primer, DNA primer. So DNA primer is the DNA primer. So it is same as the DNA replication mechanism. We need a DNA primer, but this DNA primer is not formed naturally. The DNA replication mechanism, the DNA primer is formed naturally, right? But for this one, DNA primer is we create ourselves. Kami buat sendiri punya. Okay, after that, it will elongate the DNA strand until the target that you want. So after the the process is repeated many times. We call as cycle, kami akan repeat semua proses ni until it produce thousand type of the targeted DNA lah. So yang ni is using PCR, but normally we not recommended use the PCR punya method lah compare using PCR punya method, because PCR although is very used, but the PCR normally the main kami nak is using is to amplification the gene, not clone the gene. But nowadays there are the technology that already can use PCR to clone it. So in examination normally you better use conventional method lah, culture media. So finally this is all the process that you need to use, the overall process lah, genetic engineering punya process. So finally you use this cell culture. So this cell culture already contain all the recombinant E. coli already. So this E. coli you can use in many fields. See to see whether what are the see your gene of interest lah. Kalau gene of interest is insulin, the first product insulin, maksudnya ini bacteria cell akan produce banyak insulin lah. So after several week, ini medium ni bukan sahaja ada bacteria, dia pun banyak bacteria, dia ada banyak insulin. So you can purify this media to isolate the insulin, so that insulin later that this insulin will be used in the medical field lah, and then will be purified in the pharmaceutical industry, then can be sold in the market. So this is the general process of genetic engineering. So there are 5 main stages. First stage is DNA. You need to isolate DNA and then clone. And then after that you need to cut it. Second stage is cut it. Third stage is insert. Fourth stage is transform. You need to transform into the cell, fight out, and then fifth stage is screening process. So ini semua proses dia lah. So hopefully you guys can understand it. So I think that's all for today. So for today, we only learn about the overall process of the genetic engineering only. We not sentuh application yet. So the application that you will learn is using insulin. Awak akan belajar guna insulin punya as an example. And this is the very famous topic. If you refer to past year, mainly every 2 year, 3 year dia akan keluar punya. Dia akan keluar, akan keluar, want you to write all the procedure in genetic engineering. Normally marks is 10 marks, 11 marks, and even 12 marks. So there are banyak lah. So normally you are using the insulin as example, and also you are using another one is plant and another one is thyroid growth hormone. So but the application actually is not very hard. Application normally is using this basic. Memang awak kena using this basic. But the application in insulin and also in plant, there are some minor step lah. There are some minor change of the step. But normally the theory are totally same. So if you already, so I hope that everyone can understand it, the whole process of the genetic engineering. So ini I think this is the very famous topic lah. Normally kalau memang exam question lah, especially essay, 1 year, 2 year dia akan keluar punya. I don't know last year come out not. If last year not come out, maybe this year dia akan keluar. Normally like that punya. Dia akan 2 year atau 3 tahun, 2 tahun, 3 tahun dia akan keluar. So I think that's all for today. So now already 9:22. So any question? No question. Wow, semua master this topic. Very good. Okay, so this technology actually is for you guys, for later if you discover degree in biology, normally this technology is the must lah. Awak akan awak akan belajar ini punya. Memang 100%. Kalau awak belajar biology, memang 100% akan belajar ni. 100%. Okay. Whatever biology lah, biology ke, biotechnology ke, biomedical ke, apa-apa biology lah, microbiology, genetik ke, semua memang akan belajar ni. Ini the must lah, the must technique you need to know. Okay, so that's all for today. So, any question? No question. Okay, fine. Okay, so that's all. So, thank you guys. So, see you tomorrow. Bye. Tomorrow we will go to the application part. Bye bye.